The goal of this procedure is to study breast cancer cell proliferation, colonization, and migration in a human bone tissue explant model system. This is accomplished by isolating trabecular bone tissue fragments from femoral head surgical specimens and co-culturing them with breast cancer cells expressing luciferase and green fluorescent protein. Bioluminescence imaging is used to measure proliferation of cells and verify the colonization of breast cancer cells in the bone tissue fragments. Patterns of breast cancer cell colonization are imaged by fluorescence microscopy. The marrow compartment can be flushed from the fragments for analysis by flow cytometry, bioluminescence, and staining assays. Breast cancer cell migration towards tissue culture supernatants and bone tissue fragments is measured in transwell migration chambers with bioluminescence imaging. Bone is the most frequent site of breast cancer metastasis, and ultimately, this model provides a new window for studying breast cancer cells within the bone metastatic niche.
A big advantage of this model is that it provides direct access to the microenvironment of human bone tissue fragments, allowing us to easily observe and analyze breast cancer cell behavior during short-term co-culture experiments.
This method can be used to study key questions about mechanisms underlying breast cancer metastasis to bone, with the goal of identifying therapeutic strategies to effectively prevent and treat it.
Although this model can provide insight into breast cancer metastasis, it can also be used to study other bone-seeking malignancies, such as prostate cancer.